Okay, hey everyone. So today we've got our first lecture in stem cell biology and regenerative medicine. So we're going to go through what is regenerative medicine, what is a stem cell, what is regenerative medicine, what's a stem cell, and we're also going to go through how you establish a stem cell, how you assert that it is in fact a stem cell, and also what's being done in regenerative medicine in the Cincinnati Children's Hospital. So let's move on to what regenerative medicine is. So the definition of regenerative medicine, and it's quite broad, it's designed to be really broad, is the use of stem cells for the repair, regrowth, and in some cases replacement of tissue. Now this is a definition out of Nature Journal, and you might notice that it's pretty broad, right? It's really broad in terms of it's designed to be applicable to a whole bunch of stuff. I'm just going to move the camera a wee bit here. To a whole bunch of different therapies and approaches to stem cell research. And the important thing to know about stem cell research here is that unlike, say, respiratory research, it's really cutting edge. So all of the approaches and our knowledge are changing very quickly in stem cell research. It's very cutting edge. As opposed to, say, respiratory, which has very well established background information and prior art, which allow you to perform protocols in a very refined and fully understood manner. In regenerative medicine, by contrast, our understanding of stem cells is constantly evolving. We can talk about mesenchymal stem cells, which we are just getting an understanding of now, and we're going to talk about in this lecture a little bit of hematopoietic stem cells, hematopoietic stem cells. Hematopoietic stem cells were the first ones to be understood, but we're still developing in our understanding of hematopoietic stem cells. So let's continue. So then, well, what is a stem cell, right? What makes up a stem cell? This is something that's quite hotly debated. Over here. So what is a stem cell? What is a stem cell, right? And kind of there are two critical things that make a stem cell. And those two things are self-replication and the ability to give rise to other cells. So what does that mean? What does self-replication and the ability to give rise to other cells mean? Well, it means that our stem cells, and let's assume that we've never heard of stem cells before. Stem cells are the cells that create the lineage of the 250 odd different kinds of cells that we have in our body. They sit at the top of the tree. They sit at the top of the tree, and they allow division into precursor cells, and then those can divide into all different kinds of cells, right? So they sit at the top. sit at the top of the cell differentiation tree. Now what that means in terms of the ability to give rise to other cells, of course, is that this stem cell, right, let's presume it's a hematopoietic stem cell for argument's sake, so it's a HSC, right? Now that can give rise to the hematopoietic precursor cells. I think that's right. So it differentiates into a materially different kind of cell. But in addition, it has the ability to self-replicate. And I'm going to discuss what that means right now, but I need an eraser. So we just established, right, that a stem cell has the ability to self-replicate, give rise to itself, and also give rise to other cells, right? But there are actually a lot of cells that can give rise to themselves, plenty of cells that can give rise to themselves. The liver, for example, pretty much all of the tissue regeneration that we see in the liver, and we'll discuss this later in the course. All of the regeneration that you see in the liver is actually fully mature cells just dividing, right? So a lot of cells can do that. Most cells can do that. The ability to give rise to other kinds of cells, that's the challenge. But we need to have an assay, a way of verifying that these two things are present in the cell. And we need kind of a hard test to know what the case is, right? So, how many divisions makes a stem cell, right? How many divisions makes a stem cell? So like in theory, you should have an infinite number of divisions. Any stem cell should be able to divide forever. It should be able to keep going and going and going and going and going. But in practicality, you can only observe a population of cells for so long. You can only look at it for 10 or 15 or 20 generations, depending on how long the generations take. You might be able to research them for 2 years, say for example. Maybe 2 years is like a long honours project, and that's verging on the end of reason in terms of studying how long we can keep dividing them for without having a fully devoted research grant. So, in our attempts to answer this question, we've kind of had two major approaches. And those two major approaches have been cloning cells, and we're going to talk about what exactly cloning is, but we're going to give a quick overview of it now. Cloning cells and transplanting cells. Both of which the goal was to take the stem cells and then get them individually or put them into a new situation where they can divide, and do that a number of times so that we could establish that the cells were not only giving rise to themselves, but also had the ability to give rise to new kinds of cells. They're sitting at the top of the cell tree, as it were. Right, so let's talk about the cloning approach. The cloning approach. So what did they do? In the cloning approach, I think this was, I don't remember, 96 I think this occurred. But what they did was, they were researching hematopoietic stem cells. So these are the stem cells that we get from bone marrow. There's not a whole lot of them in bone marrow, there's a whole lot of different cells in bone marrow, but some of hematopoietic stem cells exist in bone marrow and they don't exist elsewhere in the body. And these stem cells were extracted, and then, so let's draw it. So you have the bone, right? And you take your bone marrow out, right? And then you purify it as best you can to get your hematopoietic stem cells, right? And hematopoietic stem cells, and we'll talk about this later as well, but hematopoietic stem cells can be easily refined using the CD34 marker, CD34 antigen, CD34 antigen. And triple plus means that it's really strong in terms of its response, right? So you take CD34, you put it in with your bone marrow, and you get out your hematopoietic stem cells. That's the idea. HPS, hematopoietic stem cell. Okay, so we've got our hematopoietic stem cells, and we want to verify that they are indeed stem cells, right? So we need to establish that those stem cells are in fact giving rise to themselves and giving rise to other cells. So what we do is we take this liquid and we dilute it to the point that when we pipette a certain amount of that solution, we get on average 1 cell per drop, right? So that's a drop of solution, and in that drop we have 1 cell. 1 cell. Drop of solution. Right, we have a drop of solution, we have 1 cell and drop of solution. Then what we do with that is we plate it. Right, this is a plate. There's a whole bunch of different little wells. Right, might have 96 wells. 96 wells. And then we put a drop in each well. Right, 1 drop per well, which means we put 1 cell in each well, and then we let them grow and divide for 21 days or whatever it takes, such that we have a colony. Now, what's the significance of having a colony in one of these little wells? Right, so say we get a colony here. Right, what's the significance of getting that colony? Well, it means that the cells that we put in that well are dividing. Definitely dividing, which means they're definitely giving rise to themselves or others. Now remember that one of the differences between normal cells and stem cells is that stem cells should be able to divide forever. And in this test we're trying to verify how many divisions we need to be able to call something a stem cell. Right, so we take our plate, it's been 21 days, we take out our colony that has divided, and maybe we have 96 colonies, or maybe we have a dozen, or maybe we have 1. But we take out our colony that's divided, and then we replate. We replate. Replate. So we put that into another plate of 96 wells, and then after we replate 5 times, replate 5 times. We replate 5 times. After we've replated 5 times, we know that those cells are dividing. But in this particular experiment, we found that the cells we have were not able to give rise to the full regimen of cells that they should be able to. They were no longer fully pluripotent or multipotent. In this case it was hematopoietic, so they were no longer fully multipotent, right? No longer potent. No longer multipotent. No longer multipotent. Right? That's a problem. That's a problem because it breaks one of the rules of stem cells, right? Those stem cells were not giving rise to other kinds of cells, which means that they weren't, in the critical sense, they weren't stem cells. So, what do? Well, It's a very smart scientist, about 5 years after this experiment, thought that, well, maybe the issue is the niche. This is a key word. The niche. The niche was wrong. Now what does that mean? What that means is, when we grow our stem cells, they like to be in a particular place. They like to be in a particular, very special environment. So let's take a very quick example of our bone marrow here, right? So on our bone marrow, you have tissue around the exterior, right? You have another tissue in the center. I can't remember what these are called, but I'll work it out. And right in here, your stem cells like to grow. Now, that means that there's a whole bunch of cell signaling going on between your stem cells and the cells around them, right? That's going to be kind of global signaling that's occurring from various glands in the body, to cell-to-cell signaling individually, to excreted, so contact signaling, to excreted signals between cells in the immediate environment. All those things contribute to the niche of those stem cells. And if that niche is not just right, those stem cells are not going to behave in the way that you want them to, right? The niche was wrong, right? So where do you get the niche right? Where do you get it to be exactly the way it is in the body? How about in the body? Seems legit, you know? Right, so what these researchers did, and this is kind of the gold standard, the primary approach now to verifying stem cells. And what they did was they got a mouse. I'm going to do my best mouse drawing now. So, you know, it's going to be terrible. A little mouse. A little mouse. It's got ears. Right, you got your mouse, right? And they irradiated, take a step back. They extracted the bone marrow sample from a healthy mouse. Healthy mouse. Right, and they extracted bone marrow. Right? They extracted the bone marrow from the healthy mouse. Right? Then they got another mouse and they irradiated that mouse with X-rays. Now this can be kind of confusing and seems a bit arbitrary. Like why would you take bone marrow from one mouse and bother to put it into another mouse after irradiating the second mouse? Well, what's the point? What's the point? So what they're trying to do is use the second mouse as petri dish, right? The second mouse, and I'm going to draw it more poorly this time. So this second mouse here is being used as a living petri dish to grow hematopoietic stem cells in, right? So we're going to grow this first mouse's cells in this second mouse. Grow the cells of mouse 1 in mouse 2, right? And the point of that, why'd you go out of focus? There you go. Now the point of that is that we need the niche of this mouse, right? Because we remember that the stem cells that we first plated into exterior culture, into in vivo culture, were not pluripotent, were not multipotent. They lost their stem cell ability because the niche was wrong. So we need the niche in this second mouse's bone marrow cavity to grow our new stem cells. Now, why do we need to X-ray Why do we need to x-ray this second mouse? That's because if this second mouse had all of its stem cells, it wouldn't need the new stem cells. It wouldn't uptake the new stem cells. It wouldn't use them, and they would just kind of be shrugged off. And also we'd have no way of testing that the cells that we put in were in fact growing and dividing. So we irradiate this mouse. That irradiation kills the cells that divide the most, in this case stem cells, kills the stem cells, leaving us with a blank canvas, with a mouse petri dish, with a nice niche to grow cells in, right? So let's say that again. You take a healthy mouse, you give it a dose of radiation which would be lethal, because within your bone marrow you have hematopoietic stem cells, which would give rise to your blood lineage. So it takes away your ability to create new blood. And so now we have a mouse that has no hematopoietic stem cells, therefore no ability to make new blood. And we want to see if taking the stem cells from this mouse can grow and divide in this mouse. Right? So we extract that bone marrow, we create a new test bed, and then we put this bone marrow from the healthy mouse into the other mouse. And the result of the experiment is we see whether those cells grow and divide and give the second mouse the ability to survive by giving it a blood lineage, by giving it the ability to create new blood cells, right? All of that's a very in-depth explanation of what we're doing, which is trying to see if there are blood cells, stem cells in this mouse, hematopoietic blood stem cells. And so, what's the positive result? This mouse survives. If this mouse survives, we know that we have put stem cells into this second mouse. Because if we didn't, it wouldn't have the ability to create blood, and therefore it would die. So if we've successfully taken stem cells from this mouse, put them into this mouse, right? So before, it doesn't have the stem cells at the top, and it doesn't have a blood lineage, right? No blood lineage. But if we put the stem cell in, I'm going to draw it a bit more carefully, it has a blood lineage, and it has the ability to create new blood and survive. That's kind of the basic experiment of stem cells, and we're going to go over that a couple of times, so it's okay if you don't fully understand it right now. But the point is, by having this nice niche, and by taking these stem cells from this other mouse, and by seeing that this mouse survives, we know that there are stem cells in this sample. Now we do that four times. So we take the stem cells from this mouse and we replate them into another mouse, and we take it from that mouse and we replate it into another mouse, and so on. And after 5 times, we know that somewhere in that sample, in that initial sample, there were definitely stem cells. Definitely stem cells. Because after 5 replatings, we know that they had a practically infinite lifespan. They were able to give rise to themselves, and this is important, every time we put them into a new mouse, that mouse survived, and therefore those cells were able to give rise to the whole blood cell lineage. Now that's really important, and that's kind of the core definition of a stem cell. Okay, so that's kind of the intensity of this particular lecture. And that's just a very quick overview of an understanding of how we verify that stem cells exist. So, very quickly at the end, so what is this being used for? We use it for regenerative medicine and stem cell therapy, so trying to regenerate any tissues that are damaged in a person, in a patient. There are a number of research projects going on. Let me get my notes. Right. And in the next lecture, so I'll just write up what we learned today, right? So today we learned what is regenerative medicine, right? And regenerative medicine is the use of stem cells to repair, regrow, or replace any tissue of the body. What is a stem cell? Right? And a stem cell is a cell that can give rise to itself and a number of other cells and has a practically infinite lifespan. And then we went through the assay for verifying stem cellness. Stem cell. Right? And we went through cloning. And we also went through transplant. Right? And in cloning, we took individual cells and we saw if they proliferated and created a colony. And we did that 5 times. And we found that, in fact, those cells weren't stem cells anymore because the environment where they were in was not reflective of the in vitro niche that they need to grow in. And then we did a transplant approach where we irradiated a mouse, took healthy hematopoietic stem cells and put it into that mouse, saw them grow, saw that the mouse survived and therefore had a blood lineage, and then we did that 5 times to verify that those same stem cells from the first mouse were able to go into the second mouse and grow a new blood lineage. And then we took that new blood lineage and put it into a third mouse and it was able to grow a second blood lineage, and so on and so forth. And so we verified that these stem cells can be taken out of a first mouse and grow effectively 5 whole new blood lineages for 5 whole new mice. And that means that they were able to give rise to themselves and to other cells. Thank you very much, guys. See you in the next lecture.